It's good to be here. I'm amazed. I've been at Georgia Tech now for, I think, 12 years, and this is the first time I give a talk at this breakfast seminar, so I'm actually nervous. It's good to give a talk when you are nervous, and because it's supposed to get the best out of you, or maybe the worst. We'll see. So I want to tell you today about a series of projects that we've been carrying over the years, really. I mean, this is work that has been stretching, maybe I'm going to say over the period of 7 or 8 years now. And all the work that I'm going to tell you about is work that I've done in collaboration with John McDonald on, so it has to do with ovarian cancer. And this work has really branched into other projects over time. So with the things that we've learned from ovarian cancer, we've done work on prostate cancer, and now we're starting a few new things that we had never thought we were going to be working on before. And this collaboration with John also ended up branching into a collaboration with Baylor College of Medicine with Marty Matsuk, who's an expert also in ovarian cancer, and Marty's group has developed one of the first mouse models of ovarian cancer, and for reasons that they're going to be very clear in a minute, having a controlled, well-behaved, nice system for looking at disease goes a long way. Okay, we would all like to do these things with humans, but the experience with humans are only as good as the samples that you have access to, which can be quite limiting in this case. So before I start telling you about ovarian cancer, I wanted to give you a quick update and a quick preview. I am lucky to have Matt Torres and Dave Smalley here from our new center at Georgia Tech. Well, it's not officially a center yet. Let's call it a core. We call it the System Mass Spectrometry Center, or CORE. It's a new set of facilities that we are setting up in the new EBB1 building. And they're going to have capabilities in proteomics and in metabolomics to do work on both targeted and untargeted experiments. Okay, and that I'm going to tell you about these targeted and untargeted experiments in our own research. But I wanted to tell you that this type of thing that we've been doing as a research project now, they're going to become something that you could go to the facility and say, hey, I'm interested in doing this. What can we plan together? Okay, so the final space of the facility is going to be in the basement of EBB. This is not going to be ready until I keep updating this slide. Okay, so what does it say now? Okay, I just said beginning of 2016 because I don't want to update it anymore, but roughly February 2016. It's a really nice space. We're very lucky, really beautiful space in EBB. But right now we are operating on temporary space that was given to us temporarily for 6 months. The room is 2034 in EBB. And the manager of the course is here, David, so he's the point of contact, but I'm happy to help you with this if you have any questions. So we're very lucky that the Institute has invested in some instrumentation for doing this work. We have a suite of instruments. These are all mass spectrometers that are used for measuring different molecules in different type of samples. There are different kinds of mass specs because we want to determine either proteins or metabolites, and so they are tailored. It's like a bike. Okay, you can have a road bike or a mountain bike. They're both bikes, but you're not going to go with a road bike on a mountain trail. You can try, but it's not going to work. Okay. So phase one of this deployment of instruments is pretty much ongoing. The instruments are here. They're pumping down. They're being tested as we speak. And then we have a second phase of instruments that we're thinking about what capabilities we really want to add to the core as we have customers coming in and basically saying, this is what I want to do, this is what I do. Okay, so took a lot of work. Okay, so we've done some work. As a part of this development, we entered into collaboration with Thermo because we wanted to save some cash. So we saved about $1 million by entering into this collaboration. And now we are partnering also with Emory because Emory has a big cystic fibrosis center. And that center is very interested in using the course here and some of the capabilities here on things that they don't do at Emory. Okay, so just try to complement what we do here with what's being done at Emory. Okay, so with that, I wanted to give you that preview because I know that many of you may be interested in doing some of these experiments before, but I'm here to tell you about metabolomics. And metabolomics is one of the newer omics fields. And you know that all these omics fields are really enabled by new technologies, okay? Genomics was enabled by our ability to sequence genes, okay? And so it's really interesting to really look deeply into how these fields emerged, because at the very beginning, there's always a lot of research in analytical chemistry developing the tools, and then the tools become very mature, and they're taken away by biochemists and biologists, and the analytical chemists jump onto a new thing. Okay, so then it came proteomics, was really enabled by high throughput mass spectrometry and soft ionization mass spectrometers in the 90s. And that's pretty much, it's a very sophisticated, very complex set of tools, but it's doable and it's relatively routine depending on the experiment that you want to do. Some experiments are routine, some are much more complex. Metabolomics came after that, and I'm going to venture to say that it's moving faster than proteomics, because the tools for metabolomics are similar to the tools in proteomics, with the exception of the bioinformatics part. The bioinformatics part in metabolomics is still where proteomics was 15 years ago. So it's still up in the air, lots of things changing, different groups coming up with different tools, and so on. But metabolomics, as the name implies, is the system level or network level measurement of metabolites. Some people call metabolomics global metabolomics. I don't like to use the word global, because as you're going to see, we never measure all metabolites known to man. Okay, we never measure that. We cannot measure that all at once yet. Okay, so it would be too ambitious to say that we are doing that, but we're trying to get there. Okay, typically or operationally speaking, we're talking about relatively small molecules in the range of 50 to 1500 Dalton. Okay, so this involves sugars, lipids, amino acids, steroids, you name it. And this paper by the Skolnick group that was published years ago, I like this model of metabolite alterations, because Jeff's group was really talking about metabolomic alterations as a way that a chemical engineer would think about metabolites going up and down. So if you have chemical reactors with reactions that produce a certain chemical or deplete that chemical, meaning enzymes that may be up or down regulated in disease or some sort of perturbation, then the metabolites may go up or down. You can accumulate or you can deplete that metabolite. And I always thought that it's a very simple, very, very elegant model of networks and how metabolites go up or down. The metabolome has a lot of interesting information that may not be obvious in the proteome or the genome or the transcriptome. So the metabolome is much more an instant snapshot of what's happening in a given system. As such, you got to be very careful, because that instant snapshot can be altered if you mistreat your samples. Okay, if you don't process them carefully, if you take too long to process them, it's going to change, because it's really a much more instant representation. So experimentally speaking, it could be a little bit trickier than perhaps experiments with proteins or even genes. Okay, so the metabolites, you gotta be very skeptical about your results until shown otherwise. And I told you that I was going to talk about ovarian cancer. I don't think you really need to explain why ovarian cancer is important. It's really a very deadly disease. It's one of the leading causes of death in women, of cancer. Perhaps the most important thing is that it has, at the early stage, it has no symptoms. It's very difficult, if not impossible, to catch it early. My grandma actually died of ovarian cancer, and she didn't, nobody knew. Okay, so it has no symptoms. So it's only found in early stages by chance. It's only found really by chance. And if you found it early, because they happen to do surgery and they detect early stage cancer, the survival rate is very high. Okay, so if you find it early, the chances are very high. So we want to try to find it early. And we've been working with John McDonald for years now, trying to find ways of detecting early stage ovarian cancer. And ideally, what we would like to discover is a blood test. We take blood, we shoot that into some sort of instrument. Doesn't have to be a mass spec. I'll be happy with anything. And it says green or red. Okay, but that it's never black or white. There's always a range of situations. So we're going to talk about that. And like with many cancers, ovarian cancer is not a single type of disease. It's actually the most common type of ovarian cancer is called the serous, serous, high-grade serous. So when it's serous papillary, it tends to metastasize, and it's pretty much deadly if it's not caught early. But there are other types, and some of these other types are much more rare. They're not really understood if they're the same type of cancer that the serous papillary, But typically, the development hypothesis is that ovarian cancer happens because of the repetitive wounding of the ovary surface, okay, during the menstrual cycle. And during that process, there are mutations that trigger cancer. But that hypothesis is really being challenged by many researchers, including the idea that maybe ovarian cancer actually starts in the fallopian tubes, not in the ovary surface. Okay, so that's why that's important, because 30 slides from now, I'm going to show you some results that shows something like that. Okay, I'll remind you. The current standard for detecting ovarian cancer is a marker called CA-125 that it's as good as pretty much flipping a coin, if you put it mildly. And so in some cases it's useful if patients that have mutations that could, make them more prone to ovarian cancer. In those cases it's used, but for the general population, in general, it's not that useful. Okay, so many doctors don't even want to do it, because if it shows that it's elevated, it doesn't really mean anything. What you got to do is you got to really track it over time. Okay, so if you track it over time and suddenly you see it going up, that may be indication of some problem. Not necessarily ovarian cancer, but some sort of like ovarian problem. So I'm going to tell you about markers for ovarian cancer. And pretty much the community of biomarker discovery has agreed that detecting one biomarker, it's just not going to happen anymore. All the diseases for which one biomarker is enough, they're all done. Okay, we're going to need panels of biomarkers. We're looking at very complex diseases, and we're setting the bar very high. We want to do early detection. So we really want to look at molecules, a group of molecules, maybe 4, 5, 6, 10 going up or down. Okay, so that's what I'm going to be telling you about. And the reason mathematically it's very easy to understand why that has more discriminatory power. If you only have one biomarker and you have two populations, and this biomarker is not very specific or sensitive, you see in this case there's a lot of overlap between the populations, right? And it's going to work only in the most extreme cases. If you have a second marker, somebody may propose, again, along this axis there's quite a bit of overlap, it's even worse. But then you can combine these two biomarkers. You can just say, in this case it's a trivial mathematical function, it's a straight line that combines the abundance of A and B. If you combine those, you can basically say, okay, this is the dividing line, and with this dividing line I can actually separate the populations. Well, if you can imagine this in 11 dimensions, that's basically what we're doing. Okay, so you have a multivariate plane that can separate populations. Okay, that's the idea. It's a function. Okay, sometimes we call it a score. We just call it a score for simplicity. But okay, you're having breakfast, so maybe I should have warned you about that slide coming up. Okay, the next slide is a little gross. Okay, so we would like to do early stage detection of ovarian cancer, and we would like to do that in humans, but because it's so hard to find early stage samples in humans, in parallel with the work that we've done in humans, we're also working with a mouse model of ovarian cancer. And the reason why it's important to look at a mouse model is because you can follow disease progression in a very controlled way. So together with Baylor and with John McDonald, we've been working on this mouse model that it's basically a double knockout mouse model where it has these two genes, DICER and PTEN, deleted. And so PTEN is a suppressor gene, and DICER it's a regulatory gene. And when you suppress these genes in the fallopian tubes, actually what you see, this is the whole reproductive system of a mouse. Okay, this is the uterus here. These are the fallopian tubes. These two little glands are the ovaries, and in this case this is a cyst, could be benign. Okay, this is a cyst, and this is an early stage tumor that has not yet metastasized. So basically you know that typically around 6 to 8 months these mice will start developing ovarian cancer. You don't know exactly when, but if you sacrifice them you can find out if they're early stage or very late stage. So what we did is we started studying these mice, trying to look at markers in blood of these mice. And after 10 or 11 months, the tumors actually spread, metastasized, mice die every single time. Okay, so this is a pretty good model of ovarian cancer. It mimics many of the molecular features of human ovarian cancer. For example, these mice have elevated CA-125 and many other features that you would see in humans. So as far as we know, it's an adequate representation of what you would see in human patients. So what we set to do is we set to do a metabolomic investigation we said to say, okay, is there any alteration in blood from these mice at the very early stage of disease? Is there anything that we can detect that's going up or down that will be a good indicator of disease? Typically, what we're doing right now is we're applying this very complex workflow, and we had to give it a name. Everybody has to have a name for these things. We call it MUSIC. So it's a metabolome wide coverage workflow. It's very complex. So we basically take a blood sample, we precipitate the proteins, we end up with serum sample, we precipitate the proteins, we end up with a metabolite extract. You can do that precipitation with different solvents, and you end up with different extracts of different polarities. So you're really doing a fractionation right to begin with. And you can have a more polar fraction and a less polar fraction. The less polar fraction, we apply a lipidomics protocol. This is the expertise of Al Merrill's group and Cameron Stollar's group. It's basically specifically for lipids. This fraction, the more polar group, we split it in two. And what we're doing now is we're starting to do NMR. This is together with Art Edison at UGA. And we're also splitting it to do a separate type of mass spec just for the polar fraction. So you see there's a lot of work involved in this. The trick, like I told you before, comes after you combine this huge amount of data. What do you do with it? So Julia Kubanek and I had a project for 3 years, and I always joke that we collected data for 8 months, and we spent the last 2 years deciding what to do with that data because it was so much, so complex, so careful what you wish for. In that case, we actually had metabolomics, mass spec and NMR metabolomics plus proteomics data on the same system, and it got so overwhelming, okay? So careful how far you want to push these things. But you need to find a way of combining these and obtaining what we call features. Features are either pairs of mass to charge and retention time, or frequencies in the NMR, and then identify those metabolites which are going up and down. So this whole protocol is what we're using right now on systems that we already done some pilot work. In the case of the mice, we didn't start like this because we didn't know if there was going to be anything in early stage mice. Okay, so I'm going to show you the simplified protocol, which is to take serum samples, we precipitate the proteins, okay, and this is actually sounds simple, but you gotta. This is where your OCD has to kick in, because garbage in, garbage out. Trust me, if you don't do this right, everything else is a waste of time, and we've been there, okay? We've spent 1.5 years on samples that were a waste of time. And then at the end, you talk to your collaborators and you're like, are you sure that the cancer samples spent the same time in the centrifuge? And you see them pause for a minute and they're like, oh, I didn't realize that that was so important. That's it. It's 1.5 years of work thrown away, because if you don't keep things constant, and there can be any variable that will distinguish, any sort of like confounding factor that will distinguish between the two populations, and you don't want that, okay? You want to minimize that. Once you prepare your sample, you go into a mass spectrometer coupled to chromatography, typically UPLC, to separate and detect those metabolites. That's a pretty important process. So like I told you before, key to this process is the ability to take metabolites in the liquid phase and put them in the gas phase with a charge. Mass spectrometry works with ions, and we use a very cool technique called electrospray ionization that basically, this was not invented for metabolomics. This was actually invented for painting cars, okay? So electrospray was invented for spraying paint on cars, but somebody very clever said, how about we use it to make ions? And it works. And you can make ions by flowing your liquid coming from your chromatographic column and applying a potential, charging the droplets, very high voltage, okay? And that makes ions. Those ions go into a separator. In this case, we use a time of flight, and time of flight is one of the most elegant techniques for mass spectrometry. The way that it works is that if you start with a mix, let's say that we're all ions, okay? We're all holding a charge, we're all ions, and we need to be separated. We all have different masses. My mass is kind of like growing. We all have different masses, and we're kicked into this chamber, okay? We're kicked, and we're all given the same energy. But because we have different mass, to compensate for that same energy, we're going to have different velocities, okay? High mass is going to mean low velocity, and vice versa, okay? So the way that it works is that the instrument basically injects the ions, okay? Injects the ions here, and the ions start separating. So this would be heavy ions, this would be light ions. in particular, this type of instrument uses what's called a mirror or a reflectron, where the ions bounce back and hit the detector. The reason why you need this mirror is to keep the instrument compact. Otherwise, it would make it pretty long. Okay, so this is the time of flight of ion. And this process that took me 3 seconds to scroll through the slides happens in microseconds. Okay, so this is a very fast instrument. It's keeping up with the chromatography that is happening. Okay, so I'm going to go quickly through that. So you separate in the UPLC, and you further separate in your mass spectrometer. The computer hopefully is keeping up with this and detecting everything that's hitting the detector. And you're going to end up with this bidimensional data, which is chromatography in one axis and mass spec in the other axis, okay, for each sample. You're going to extract from each one of those, from that data, you're going to extract the peaks. And you're going to align the peaks. So you want to make sure that you're comparing apples with apples. That step of extraction and alignment and integration, it's also where all the problems happen. So it's full with pitfalls. Very important that it's done right. But once you end up with this massive spreadsheet of features, we call them. We don't even call them metabolites because we don't know what they are. You need to start figuring out what those are. If you go to a conference where somebody's talking about metabolomics, some people will claim, oh, I am detecting 1 million metabolites. That is wrong, okay? They are detecting 1 million peaks. Those peaks may not be metabolites. They may be contaminants from the VWR methanol, contaminants from your column, okay? So you got to be very careful and curate that data to make sure that those peaks are not known contaminants, those peaks are not in the blank, and so on. Okay, so be very, very careful with that step. But once you go over that step and you're sure that the data is as clean as you can, then you're ready to go and talk to your bioinformatics colleagues and start playing and trying to see, is there a function or is there a set of metabolites that I can use to separate, let's say, early stage from control mice? Or the answer may be no. Sometimes there's no way of separating it. Sometimes it doesn't work. Okay, so we curate the data, and then we do multivariate analysis. And then once you know those metabolites, you're going to identify them, and I'm going to show you how to do that. And then you need to map them back into pathways. Okay, so you go from blood all the way to pathway mapping. So let me just show you some results for mice. So the first thing that we did is we ran a set of mice through all this workflow I just described. And we had early stage mice, and we had late stage mice and control mice. Okay, so early stage mice are the red symbols, late stage are the green ones, and the blue squares are the controls. If you take all the data that you get from the mass spectrometer and you do something relatively simple, such as principal component analysis, and this is the PC1 versus PC2, this is what you get. You see that the late stage mice separate relatively well from both the early stage and control, but the early stage and control don't separate. Is this surprising? No. I told you that early stage has no visible symptoms, so overall it's very hard to detect. Okay, so overall it's very hard to detect. So this was a little disappointing, but we're like, look, wait a minute. First of all, PCA is trying to capture the things that are the most different. Okay, so late stage is so different, these mice are extremely sick, that it's obvious that that's different from the other two sets. Okay, so that in itself confounds a little bit the analysis. So what we did is we took away the late stage samples from this data set, and we stuck to the early and controls. And instead of using PCA, we used what's called a supervised method. So we basically split the data in two. We used a training set and a test set. The training set builds a model, builds a function that separates. And the test set, we pretend it's an unknown, and we see if we can classify it. Okay, so that's the way it works. And we used all the data for this. Okay, we are talking about a set of 934 features. Like I told you, I mean, there are more than 934 known metabolites. So by no means we're looking at the whole metabolome. Okay, it's a good fraction, maybe I'm going to say a tenth, depending on what you know. But this is all negative ion mode data, so we're specifically looking at molecules that can deprotonate. Okay, so let me show you what happens when you do this in a supervised fashion. So we first train the model, and of course, when you train the model, It works very well. You can separate early stage from controls perfectly when you train the model. Okay? And this is using all 934 features. But then when you test this model, it doesn't work so well. You see how there's some blue squares over the decision limit and some red circles below the decision limit. So that means that we're misclassifying mice. And you don't want misclassified samples because it means that you're either telling somebody you have ovarian cancer when they don't, or you're telling somebody you're fine, go home, and they have ovarian cancer. So none of those situations is really something you want to be in. Okay? So it's pretty challenging to do this. So what can you do? Well, computer scientists have figured this out before. Okay? So in the same way that when you go and you open Netflix, Netflix kind of knows what you want to watch next. And that's, I'm not joking, actually the person that invented that, it's the person that did this work with us. Okay? So a faculty member in computer science, Alex Gray, he's a millionaire now because he figured out the Netflix thing. Yeah. But this is called machine learning. Okay? So you can teach these models how to learn what are the best metabolites for separating these things. Okay? So one way of looking at that is by using what's called the genetic algorithm. And I'm sure many of you know about genetic algorithms. If you've done some genomics work, it's used all the time. The idea is the following. We're going to take subsets of these 934. Let's say we're going to take sets of 90. We're going to make random sets of 90, let's say 200. And we're going to try them. Does this set work to separate? No. Yes. Maybe. So you're going to rank them. And you're going to see which sets work best and which sets don't work at all or are mediocre. You rank them. You throw away the bottom half. And you keep the best. You shuffle them, and you shuffle them using techniques similar to genetic mutations. So you have mutations, you have crossover, and things like that. And you start over again. So you start with sets of 90, and you end up with smaller sets, but that work much, much better. So this process is actually repeated over and over and over again. It can take days to run. Okay, so we did that with our mouse data. And we came up with a new set. So this here is the new set. So we went from 934 features to an optimized set of 18 features. In this case, we can probably safely start calling them metabolites that actually separated well not only in the training stage, but also separated, in this case, exactly separated in the test phase. The reason why it works so well even in the test phase is because these are mice. So these are relatively homogeneous populations of samples. Okay? I'm going to show you work with humans. It doesn't work so well. Okay? But in mice, sometimes you can expect this. And this is all nice and great, but now we need to know what those 18 are because nobody wants to predict on peaks. People want to know what are these molecules and do they make sense? Because if they don't make sense, then we need to start over again. Make sense means they do map into biological pathways that are known to be altered in cancer, and particularly maybe in ovarian cancer. Okay, so how do I identify these? So this is, if you've taken an organic chemistry class, you go back to your organic chemistry class. You look at your chromatograms. This is one for a control sample. This is one for an early stage sample. And it would be very tempting to start to look at peaks and small differences, but careful because you're not just interested in differences. You're interested in differences that are correlated with early stage cancer. There could be many differences, okay, between these mice, but you just want to look at the main differences. As a matter of fact, if you look at this plot here, you see that, for example, early stage mice in this direction have a lot of variance, but this is not correlated to early stage disease. Okay, so careful because this is not the variance that we're interested in. We're interested in this orthogonal variance. Okay, so you look at the chromatograms and then you look at the specific peaks that showed up as changed. I'm just using one here as an example. We saw that this mass to charge ratio of 452.2779 was altered. You see here two traces, one for control in blue and one for early stage in red. And then you look at the mass spectrum. So you're really drilling into the data. You look at the mass spectrum under this, and you see, okay, this is the peak that is changing. And with this mass here, this accurate mass, many times you can obtain an elemental formula for that molecule. And that reason is why we rely on really high resolution instrumentation, because we want this number to be very reliable and we don't want any peaks to overlap. If you have overlaps, you may be looking at 2 molecules at once, and that's going to distort all the picture. Okay, so that's why we need high resolution measurements. Furthermore, the mass alone is not enough. What you do is you select this molecule. The time of flight has a way of selecting a specific ion, and you fragment it. You break it into parts, and you measure the masses of the fragments. And then you start putting this puzzle together. If you're lucky, you're going to be able to search this in a database. But databases for metabolomics are not there yet. They're growing, but they're not complete. So sometimes you may search a database and get a match. That's great. But if you don't get a match, you're going to have to look at what potential molecules match this elemental formula, and then see if that molecule would match this tandem mass spectrum. Okay, so it's a pretty tedious process. So it's so tedious that you're going to understand why I show these tables. So they teach you in kindergarten of PowerPoint not to show tables. This table took us a year of work. So I am going to show it to you, like it or not, because it was so much work. Okay, we're very proud of this table. So by no means I want you to remember everything in this table. What's really important is here in this column, and these are identities of these molecules, metabolites. They have different degrees of confidence. Some are probably wrong, some are probably right, because we cannot do on all of them. Some of them are very low abundance, and on those you really cannot do reliable fragmentation. Okay, so, and some of them we have fragmentation and we have masses, but we cannot do chromatography well because there are no standards. Okay, so the ones that are bolded, for example here, one molecule that was really interesting was bilirubin. Okay, bilirubin was changed in this panel. There are many lipids, phosphatidylinosytols, some bile acids, and some triglycerides, but there's some molecules, for example, Na. Okay, Na. So this is by no means completely done. Okay, but if you, with the tentative IDs, if you go back and you map this into pathways, you see really many changes. You see changes in lipid metabolism. By the way, blue means increased in early stage. Red means decreased in early stage. You see changes in the heme catabolism. There's bilirubin here downstream. It's well known. We had detected this before in late stage, altered bile acid metabolism. So lots of things going up and down. Okay, so this is really interesting, but doesn't really answer all the questions. It's powerful because it means that there's some sort of signature of early stage ovarian cancer in mice, but we are not going to take this to the clinic yet, right? There's a lot of work to be done. And one of the things that we particularly are very interested in is how do you really improve this identification process? How do you really improve it? And that's important because any biological conclusion that comes after this is based on a careful ID. And you'll be surprised how many papers are out there where the IDs are treated very lightly. Okay, you shouldn't do that. And one of the things that we're trying to do is to measure some other property of the molecule. No, we measured the mass, we measure the fragmentation. Those are intrinsic properties of the molecule. We measure the retention time in a column. That's not really a very stable measure because it changes between different labs, even between different batches of columns. If you've done HPLC, you know what I'm talking about. You can have, you put a new column and the retention times change, so it's not very reliable. So we're working on what other things can we measure, let's say for this or any molecule, okay? This is general for any molecules to improve identification. And we're using a technique that is not so well known. Well, you've probably seen it, but you don't realize it. It's called ion mobility spectrometry. So if you've gone through Hartsfield-Jackson, you've actually been searched with one of these or even seen one of these. So I don't know if when they do the swabbing on your fingertips, and they're looking, you guys know what they're looking for. Explosives or narcotics. So every time I go through Hartsfield-Jackson, I'm ready to say, look, I'm a chemist. It's okay for me to have chemicals in my hand, which is probably not okay. But ion mobility instruments are basically the instruments back there, okay, in that corner. That's what they use. But they can also be combined with mass specs. So an ion mobility is like a gas phase electrophoresis. Okay, so you make ions, and then those ions fly down a tube, and there's a gas that flows against the ions and separates them. What's intrinsic about this process is that molecules are separated based on the collisional cross-section. And that's the shape of a molecule in the gas phase. Okay, and that's very intrinsic of a molecule, and it doesn't change between labs. So if you can measure that, and you can match the collisional cross-section, you can have an additional way of saying, yes, this is the molecule I think it is. By the way, these instruments, this is called a drift tube ion mobility spectrometer. These were developed back in the 60s at Georgia Tech in physics. Okay, and then they made it all around the world, and they're even commercial. Okay, but the theory for this is what's called the Mason-Champ equation. The collisional cross-section of an ion is basically proportional to the drift time in this tube, proportional to the electric field in that tube. So this is like an electrophoresis, and it's inversely proportional to the length, right? So the E over L is really what matters. So we're trying to find ways of using omega as an additional parameter for identifying metabolites. And this is something that we're building right now. So we have one of these instruments. It's an Orbitrap mass spectrometer. This is a very cool type of mass spec. I'll tell you more about this in a second. But we're combining the mass spec with an ion mobility. So after the chromatographic separation, you get ion mobility separation, and after that, and a collisional cross-section, and after that, you get the mass to charge, and after that, you get the fragments. So now you have retention time, collisional cross-section, mass, and mass of the fragments for ID. It's much better, okay? Coming back to the Orbitraps are really changing the landscape in terms of mass spectrometry, and if you paid attention, the new facilities in EBB are going to have Orbitrap mass spectrometers. Orbitraps are Fourier transform mass specs. So basically, you take your ions, you shoot them into this Orbitrap, and this Orbitrap, to give you an idea, is about this big. Okay, so it's not a monster instrument, the mass analyzer. So it goes into the Orbitrap, and they oscillate, and this oscillation frequency is proportional to the mass to charge. So if you measure the oscillation over time, you apply the Fourier transform to that, you get the frequency of oscillation. That's the mass spectrum, basically. It's very direct. Okay, so these are very high resolution instruments, very high resolution instruments, and can separate very closely related molecules. That's why we want them. Okay, so in this case, where you have only one kind of molecule, you get a sine wave. But if you have a mixture, you get a mixture of sine waves that you deconvolute using a fast Fourier transform. This whole thing happens behind the scenes. Okay, so you don't have to do this manually. Okay. So I show you this list of metabolites, and the question is, these are in blood. Are these coming from tissue, or this is related to the tumor? What is the connection with what's happening in the ovaries? So the other thing that you can do in these experiments is you can look at the tissue itself, but you don't need to homogenize and extract the tissue. You can actually go and do mass spec directly on the tissue. So there are many ways of doing mass spectrometry imaging. Our core facility here in IBB is very good at doing MALDI imaging. MALDI is a technique that works with a laser and a matrix. So the laser shoots on the tissue sample, and for every spot you get a mass spectrum. So if you raster the laser, then you get for every spot a mass spectrum, and then you can say, okay, build me an image for M/C 452. And it's a virtual image. It's not really an image, but it will plot that mass over X and Y. So you can do that routinely these days, okay? MALDI is very powerful, but that's not the only way. For example, there's other techniques like SIMS, secondary ion mass spectrometry, which is used actually, Georgia Tech has a SIMS instrument. It's used for materials, for imaging materials. But there are other techniques that are faster and offer complementary information. We use a technique called DESI. DESI is very interesting. I call it, it works like a pressure washer. If you pressure wash your driveway, you're shooting a liquid jet at the tissue sample. So you're shooting a liquid jet at the tissue sample, and the droplets are extracting metabolites from the surface and bouncing into the mass spectrometer. Okay, so it really works by a mechanism that's very different than MALDI, and that's why they provide kind of complementary coverage in terms of metabolites and things that you can see. So let me just show you results. Okay, so we typically apply several techniques. We take a tissue sample. We use the core here, histology core, to take a slice of that tissue sample with a cryomicrotome. We put it on microscope slides. We thaw it in a desiccator. Sometimes we wash it, depending on what we're looking for. And then if we're going to do MALDI, we use the IVV core facilities here. They've developed a system to deposit the MALDI matrix. And we use the MALDI instrument in the facility to image by MALDI. But we also do DESI in our labs, and DESI doesn't require the matrix. It works a little bit differently. Okay, so we are going to have two sets of images. This is what the whole reproductive system of one of the DKO mice, the mutant mice, on a MALDI slide looks like. Okay, so it's white because it's been already coated with the MALDI matrix. The MALDI matrix is a compound that absorbs the laser radiation. Okay, I don't know if you can see it. This is the uterus, one fallopian tube here, the other one here, one ovary here, another ovary here, a big late stage tumor here filled with blood actually, and there's a little early stage tumor there. So we actually sacrificed many animals until we found this particular sample because we wanted a sample where we could see a comparison of early stage and late stage in the same tissue. So we got very lucky to find this one. And we imaged this both in positive and negative ion mode, but I'm just going to show you one set of data. We have too much data. The software part of this is not so simple. It's quite tricky. So we've been working with Mei Wang for quite some time now on developing software to take the image, to take the data from the mass spec and assemble an image. So we came up with this software tool called Omnispect. This is actually online, so you can upload your mass spec data and it will make the images for you. You say, I want to look at a specific ion, it will make the image for you. So you don't have to have a very powerful computer to do this yourself. It's web-based. It's open source, so you can modify it if you so wish to do. But the main thing that I like is that it can do quite a bit of multivariate analysis. It has a routine called NMF, nonlinear matrix factorization. It's like a PCA for images, basically. And it can show you what are the main ions changing in the image. Now, you generate a lot of images. You generate one image for every M/C. So if you're going to look at these images manually, it's going to take you days. Okay, you're going to have to go image by image and be like, oh, this is interesting. Let's set it aside, and so on. So this will do that for you. Okay, and it works for any soft technique. So some images. This is the reproductive system. I don't know if you can tell that it follows the shape. And what I'm plotting here is the NMF image. Okay, so this is the ions that are changing the most, not a single ion. Okay, so you see that there's quite a bit of ions changing in this tumor, that those overlap with some things changing in the uterus and in the early stage tumor. Okay, so it's not a simple picture. It's not black or white. Things seem to be changing in many parts, which goes back to the theory that maybe things don't necessarily originate just in the ovary. Maybe there's some microlesions that are maybe migrating to the ovaries, and that's what's contributing to the development of ovarian cancer. And these type of images are called component images because they really combine several ions at the same time. But you can also do just a region of interest image where you just select this part of the data and you say, okay, show me all the ions that are here. So if you want to look at that, all these ions in this particular tumor region are shown in this mass spectrum, and you go through the same identification process I was telling you before. These, by the way, are all lipids, which is not surprising. Okay, I'm going to show you a list of another list that we're very proud of because it took a long time what those are. I told you that we use two techniques, and they're really complementary. Okay, so this is a MALDI image that we obtained with Cameron Sollards in the IBB facility, and this is a DESI image. And you see some obvious differences. Look at the tumor region here and the tumor region in the DESI image. So they're really different. Okay, so there's some other differences. For example, if you look at the low mass range, the DESI data is much better at the low mass range, where MALDI tends to have a lot of noise because of the matrix in that range. So you see that ions here that you don't see with MALDI. So we use both kinds of data, okay, which makes things a little bit more difficult but more complementary. On the other hand, look at the MALDI data for the ovary. So the ovary and the early stage tumor are lighting up like crazy in MALDI. So there's things that we see in MALDI that we don't see in DESI and vice versa. All right, long list, but what's important here is that these are the main species that we see changing in early stage tissue. They're all lipids. The ones that I'm highlighting here actually are also changed in blood. Okay, so we went back to the blood metabolomics data set and we looked for those. So there's a connection between what's happening in the tumor tissue and what's happening in blood. For example, this one was one that was picked for the panel. So bilirubin is also something that shows up very clearly in the tissue as being altered. Okay, but they're not a single way of showing these alterations. That's what's interesting. There are many ways of showing these alterations. There are many ceramides. There's a cholesterol sulfate there, which we've seen some similar molecules in the other panels. So it really matches the metabolomics data. But just to be true, these, look at the number of peaks that we're seeing in these images. We're never detecting 5,000 signals. Okay, so these imaging experiments, again, they're not global. When anybody uses the word global, you know that that's not there. Okay, so we're only really looking at hundreds of metabolites in these experiments. So you do need to use complementary techniques. So we are really gearheads, and we're really interested in developing a new technique. And we've been developing a new technique for imaging. We use this a third way of making ions, which is a plasma, an electric plasma, a spark, and a laser. Okay, so this is not ready for prime time in terms of using precious samples, but hopefully if, in the next 12 years, Collie invites me to give another breakfast talk, this will be ready, okay? But we're working on this. This is all nice and great from the academic perspective, but at the end of the day, what we want to do is we want to do diagnostics in humans, right? And so far what we've learned is that, yes, there's hope in the metabolome for looking at changes in early stage ovarian cancer, at least in mice. Okay, but how about humans? So the work that we've been doing for a long time with John McDonald is really directly on human samples. So the challenge is to obtain enough samples so the study has enough power to be believable, okay? Because nobody finds early stage ovarian cancer. So we have been collecting early stage samples for a while, and here you see the largest data set so far, and the paper got accepted this morning actually. We have had about 46 early stage samples, and we age match them to controls. They have to be age matched so you don't distinguish by age. And these are the details in case you're interested, but they're all stage 1 and 2, meaning that it has not metastasized. So this is early stage. Okay, so we did, this is details on the age distribution. I'm going to save you all the details, but we did very similar experiment to what I showed you for mice. We did all the metabolomics platform. And I'm also going to show you the negative ion mode data, although we also did other types of data. And it works. Okay, it works very well. So if you look at all the metabolites, all the features, it doesn't work very well. So this plot here, which is the most important, shows you the number of features that we are using. And this is the percent accuracy, if you want to put it away. So if you use all metabolites detected, the specificity and sensitivity are 60%, which is close to flipping a coin. That's not going to work. But then if you use a feature selection mechanism like a genetic algorithm, in this case we use a more sophisticated approach. We use something called recursive feature elimination support vector machines. These are actually much more sophisticated because in human data the variability is much more complex to model. And you let it select, there's an optimum number of features around 15 right there where you can discriminate very well. So if you take these subsets here, you see you can discriminate between control patients and early stage cancer in human samples. Okay, so this is what we want to do. Are we ready to take this to the clinic? I'm a pessimist, and I'm going to say not yet. So don't try to say that we have a diagnostic test, because it's only 50 patients. I would like to see another study to validate this with, let's say, 200 patients, but it's hard to find those samples, so we're trying to do that. The metabolites involved, again, they go up and down, okay? There's a change in both directions, and we spend a ridiculous amount of time also validating this model to make sure that we're not fooling ourselves, so we try different types of classification. We try different types of what's called cross-validation. Cross-validation is interesting. You take samples in and out to pretend that you don't know what they are. You try to predict them and you say if you did it right or not. And you go back and forth. And you do that over and over again. And the accuracy ranges from perfect in the most optimistic case to around 90% in a more realistic case. Okay, we want to get better than that because ovarian cancer is really a low prevalence disease. So you don't have the luxury of failing when one case shows up. So you really want to have a specificity and sensitivity that are above 95%. Okay, that's really where we want to get at. In terms of what the metabolites are, again, this is many NAs. Okay, there are many things that we could. In about 5, we spend months and months and months trying to figure out what they are. We just don't know what they are yet. But many of these are lipids. So there's ceramide here, and so on. But you have you have many interesting molecules. You have, for example, some small peptides. You know, is this going to hold for a larger population? We don't know yet. Okay, we're going to have to look at a larger population and see if the same markers hold, because sometimes these markers can help you discriminate 1 or 2 samples, but not for a larger population. Okay, so this is where we are right now, but you know, it's very promising, and we're very, very excited about this. So with that, I'm going to finish. This is the group right now. The work that I showed you, imaging work, is mostly by Marty Payne. The ovarian cancer human work is by Dave Gold, who's going to be working with Dave Smalley in the course. So he's going to be working on the metabolomics part of this. And then the rest of the group, which is getting a little bit too big at this point. And thank you very much again for the invitation. Yeah, so, you know, my limited experience with NMR is that it really highlights a different part of the metabolome. So, for example, the work that we published with Julia in PNAS, I think it was last year, it turns out that the metabolites that we were seeing in NMR, we were not seeing in mass spec. We were not detecting them, okay? And that's because there's a lot of overlap and suppression. These mixtures that you're injecting, they're so complex that some of the poor ionizing metabolites just don't show up, but NMR picks them up. And so it was really surprising. But then if you look at pathway-wise, they tend to map into the same places, which is really interesting. But the challenge is really to join the data. So it's really the frontier where people are really working on how to join NMR and mass spec data, and not just from the pathway perspective, but also even from the simple classification perspective. There are 4 papers out there, I think. That's what people are. I was reviewing an NIH proposal last week. Everybody wants to do that.
Are you seeing any evidence that the phosphoinositide, that looks like it's one of your markers, might actually be a phosphoinositide phosphate, that either while the sample is being collected or in source, dephosphorylating so that the mechanistic species?
It's not, you know, yeah, that's a very good point. I don't know the answer to that, but I need to look at that. So it's like an in-source product. Yeah, it could be, because they're very, very fragile. Yeah, they're very fragile. No, it could very well be that's the, you know, the way that we select the ionization conditions is typically a compromise, because you know, you're not looking at a specific metabolite, and you can optimize for a specific one.
Phosphatase inhibitors, the tubes you collect your...
They're not. They're not. Yeah, they're not. Right. No, they're not. And you know, you ask your collaborators what they've done with your samples, and you tend to believe them. But yeah, no, but that's a very good point.
Nice talk.
Thank you.
I was wondering where you showed the 90% specificity sensitivity. Is it for both stage 1 and stage 2? Is there like a better correlation for stage?
Yeah, that's a very good question. So we tried to do that, but you know, the answer is you don't have enough samples to do that. It's very tempting to do that, but and you can do that for other stages too. But you know, you go from 46 to about 20 samples, and then you split that into a training and the test set. I don't want to go there. I mean, you start making hypotheses of things that may not be true. So you can separate the stages. Let me just say that yes, you can separate them, but the staging is also not necessarily a science. It's done by looking at the tissues. Something that is called stage 2, some other person could call stage 1. So, you know, you need to assign that class in a very careful way, which pathologists don't necessarily do, you know, or care to do, to distinguish so well. But we would love to do that.
Yeah, actually, to that point, I mean, is it known that the tumors that kill are the largest tumors? Or like when you're doing your separation of stage in the mice, yeah, like what is your methodology for sort of staging these tumors?
So the stage, my understanding, okay, of this is that stage 1 and 2 are basically classified when they're not metastasized at all. Okay, so, but after that stage 3 is already metastatic and it has spread into the abdomen. Okay, so if I show you some of the pictures of late stage in the mice, I mean, these mice have developed what's called ascites, so these big sacs of blood, and they die irreversibly. So what kills them? The cancer has spread everywhere.
I guess just when you were comparing in one mouse between the early stage and late stage tumor, if that's a mouse with metastatic disease, how do you know which of those tumors or both tumors or neither tumor? Like, how did you sort of say, like, obviously one tumor is larger than the other, but is that necessarily?
Well, yeah. Okay, so I understand your question. So when we call it late stage, it was always metastatic. Okay, it's not tumor size. It was metastatic. It was spread. There were tumors in other parts. Yeah. And
Yeah, so for your clinical application, just to kind of understand, for humans, you only look at blood sample or do you look at tissue?
blood.
It's only blood.
Yeah, that would be blood. And the way that we envision it happening, it would be that you would collect a blood sample in the clinic, and this would be sent to a clinical lab. Okay, and the way that the work will be done is the same way that neonatal screening is done for kids, where metabolites are measured routinely. So, a panel of, like, depending on the state, but a panel of 20 or 30 or 40 metabolites, the techniques are just very streamlined. You could do the same thing once you have a panel that is reliable and you know the identity of those molecules. You could just set, you don't have to change it.
So
Yeah.
your limitation is because the detection of early cancer has to be when a person is in an operation, right? Is that why?
No, I mean, it would be, the way that we did it is, these were women that went to surgery, so blood was taken before surgery, and they had surgery, and they discovered during surgery that they had early stage cancer. But in a practical sense, we wouldn't need to do that. We just take a blood sample. That would be ideal. But are we there yet? No.
In prostate cancer, they fine-tune their diagnosis with a Gleason score, where they take cores, and it's a little more accessible to the prostate gland. Is there devices being designed to be able to take core samples?
Yeah, I don't know the answer to that, but I can answer something else. We've actually done the same work that you show you here for prostate cancer, and it works really nicely. And we were very lucky where the panel, we have a very high success of identifying what the molecules were. But so we did that first work, and we showed it to a clinician, an expert in prostate cancer, and the clinician was like, this is really interesting. Now we need to fine tune it, which is exactly what you're saying, to see if we can predict outcomes. And the other problem is that it's unclear many times. They go through the first biopsy, and it's unclear if the patient needs a subsequent biopsy sometimes. They do multiple biopsies, and those biopsies carry many risks. So we're actually working on that, where we're trying to predict what patients need second and third biopsies based on what we see in the first biopsies and the initial profile. No, I agree with you that prostate cancer, I think it's more manageable because there are different pathways for treatment. It's kind of a little more standardized than this.
Kind of going off that question, how specific are the markers you found for that particular cancer you're studying?
Yeah, most likely they're not. So many of these have to do with inflammation.
Right.
Okay, so so you can envision a scenario where somebody may have. So when we look at humans, we basically try to include that diversity in the training set. In an ideal training set, you would have a population that has all sorts of issues that could confound the results, and you will try to tease apart just metabolites or metabolite patterns that are associated with ovarian cancer. I don't know the answer to that question in our set because it's about 50 people. So they had other inflammatory processes going on because they were going for surgery, but the control population also had some benign ovarian conditions. Okay, so that's the best thing that you can do to ensure that you mitigate that risk. So to design your experiments so the normals are not completely healthy, but you also have some benign conditions that may have some inflammatory processes going on. But you're absolutely right. Are they specific? I'm not going to say yes. I mean, we don't really know that. Hopefully. All right. Thank you.